Seattle. You can correct me if I'm wrong.
Yeah, correct.
Although he's based in New York at Sinai, which is a fantastic research institute as well. So Sam, we're very happy to have you here with us.
Great.
And I look forward to your presentation.
All right. Good morning, everyone. My name is Sam, and today I'm going to present on cell-free DNA sequencing detects breast cancer in majority of newly diagnosed women in Ghana. So the incidence of cancer worldwide, in Africa, it's low when you look at sub-Saharan Africa compared to America and then European countries. But then you could also see that the mortality rates are quite higher in sub-Saharan Africa. Now, this could be explained by lots of factors, such as poverty, which definitely drives an individual's ability to seek for early treatment or even go for diagnostics. In this map, I tried to summarize the cases of cancer in one particular hospital in Kumasi, and you could see an increasing trend. And in the report, they stated that breast cancer was the top cancer in that particular hospital. I also wanted to mention that cancer arises from mutations in the cell. So we're all born with normal cells, but then as we grow and age, they accumulate mutations, and they could form tumor. And then as it grows and accumulates more mutations, it could spread to other parts of the body, such as the bone, the kidney, and the lungs, and it's called metastasis. Two types of mutations are known to be associated with cancers: somatic and germline. Somatic is what happens as you grow. You tend to kind of like acquire somatic mutations. I also wanted to explain that we could have point mutations, which basically affect specific bases of the DNA. And then you could have structural variations. An example is the copy number alterations, which is basically losses or gains of segments of the chromosome or the entire chromosomes. You could have inversions, you could have translations, and changing of the position of the chromosome. So this is a karyotype, which is kind of like a catalog of the chromosomes of cancer cells. So normally, each chromosome is supposed to have like 2 copies. So here you could see we have like 3 copies of chromosome 1, and sometimes you could have segments of chromosomes joining. So I think this is chromosome 1, and then so this is a chromosome, which is kind of like a fusion between chromosome 1 and I think chromosome 18 or so. So there is a translocation of segments of the chromosome. And you could also have high ploidy. So ploidy basically means the average copy number in every cell. So you could see instead of 2, you have more than 5 copies of this chromosome. This then has high ploidy. So copy number alterations are part of the somatic mutations that happen in cancer. Now, cancer cells are also able to release DNA into the blood, and you could actually sample this tumor DNA and look at the methylation patterns, look at the DNA integrity, look at what kind of mutations are being carried by this tumor DNA. This is a developing technology. It's very good, especially for cancers which are very invasive, and it's also good for mildly invasive diagnostics. You could actually use it for monitoring treatments because you wouldn't want to take biopsies every time. You want something which is very convenient. So cell-free DNA approach, it's one of the important things you could actually do in diagnostics and treatment. So there were some few motivations behind this study, aside the high incidence of breast cancer in Ghana and other African countries. Most of the women present late to the clinic with large tumors, and definitely there are some deficiencies with mammography. and the lack of experts and access to equipment to actually do the molecular subtyping of the breast cancer, it's also was an issue. It's still an issue. And I even heard that sometimes pathologists could miss the tumor, so giving rise to false negatives. Now, tumor biopsy is also very invasive. You could imagine going to the hospital as a woman and being told they want to take like a biopsy from your breast area. And it's also very impractical to do repeated sampling. And definitely new tools, new analytical tools needs verification and validation in different cohorts because they tend to behave differently when you move them from places. So in doing this study, we asked ourselves whether we could detect cell-free DNA from the blood of patients in Ghana to see if our methods work. This is actually a pilot study. So our specific aims was to quantify tumor fractions of cell-free DNA from these patients, and also to determine the copy number alteration profiles and diploidy of breast cancer patients, all from cell-free DNA. So this is the approach that we use. I mentioned that it's a pilot study, so we took 15 samples from the Ghana Breast Health Study cohort, which was a very big study, large study carried out in Ghana, where they recruited over 1,000 cases and 2,000 controls. But then we took just 15, and then they had plasma cells, plasma samples, sorry. And then we extracted the cell-free DNA, and then we sequenced it on two platforms. So we did the whole genome sequencing, which gives us more information, more comprehensive information about the genome. And then we did the ultra-low pass sequencing, also known as shallow sequencing. But then there are reports that the information you get from ultra-low pass sequencing, there is a high concordance between the information you get from ultra-low pass sequencing and then whole genome sequencing. Whole genome sequencing is quite pricey compared to ultra-low pass sequencing. So our objective was to be able to confidently say we could actually detect cancer. Sorry, we could actually detect cancer using the ultra-low pass sequencing platform. And then we did, the bioinformatics analysis, which was one of the reasons why I came to the Fred Hatch to do the analysis and learn from the developer of the tool. So basically, we map the reads to the reference genome, and the relative abundance of the reads in different parts of genome. We could use it to infer the copy number profile. So I'll explain in the next slide, actually. So this is an example of the computation analysis pipeline. After we did the sequencing and got the files, the FASTQ files, we took them through pre-processing methods. We mapped the reads to the reference genome, in this case, the human reference genome, and then we trimmed it a little bit. We did some recalibration or according to the GATK best practices, and then we have our files ready. And then we use this software called ICOCNA, which actually helps us in the estimation of the tumor fractions and also to look at the copy number alterations. I made mention that you could actually use the relative abundance of the reads at specific parts of the genome to infer the copy number alterations. But then there are some things you need to really correct for, and that's what we use the software to do. An example of the things you basically need to correct for are the GC content, because some areas of the genome tends to have high GC content, and those places are quite difficult to sequence. You tend to have low information or low coverage. There are some areas of the genome which are very easy to map your reads to, and there are some areas which are very difficult to map your reads to. So we did all this correction, which came as part of the software. So the software is called ICOCNA. It is an R package, could be run on the command line, and you could also do the analysis on cloud resources. And I made mention that it predicts tumor fraction and copy number alteration. was developed by Dr. Gavin Ha, who is currently at the Fred Hatch Institute, and it's freely available on GitHub. You could download and even look at the reference material, and it has quite good documentations if you want to go into it. So this is an example of the commands that we used in running the analysis. Do you want to go more into detail, but this is just an example. Great. So this is the interesting part. This is what we found when we did the analysis. So remember, I made mention that we sequence using the ultra low pass sequencing, a shallow sequencing method, which is relatively cheaper compared to the whole genome sequencing method, which is quite pricey, but then also more comprehensive. And our aim was to be able to confidently say, hey, we were able to do our detection with this method. So I forgot to mention that ICOCNA, when it was developed, had lower limits of detection. So what this means that beyond this level, your confidence of actually detecting tumor, it's low, and anything you see, you have less confidence in reporting. But then when we did the analysis, we were able to see that more than half of the patients had tumor fractions more than 3%, 0.03, mentioning could be a fraction. So more than half of the patients from both instruments had tumor fractions exceeding 3%. Okay, so this means we could actually use both platforms to see the tumor fractions from the cell-free DNA, which is very interesting. And then some also had tumor fractions more than 10%. So we could actually prioritize these patients for treatment and other things. So there's an example. So to be able to confirm the agreements between the two instruments, we did correlations actually, and then we saw that they were highly correlated. The tumor fractions from these two equipments were highly correlated. So here I want to show you briefly what the copy number profiles are. So these are basically two patients. And the various beans are the chromosomes. So chromosome 1 to X, chromosome 1 to X, and then you could see some amplifications, which are kind of like high increase of segments of the chromosome on the entire chromosome. So you could see amplifications here. You could actually see deletions at specific parts of the genome. You could see copy neutral. It means both copies of that particular chromosome are there. So this is when the person doesn't have cancer, everything is flat. You could see everything is flat. There are no amplifications or deletions or anything. So yeah, very informative. We did the software also helps us to also predict the ploidy, and it's generally between 2. But then we could see some differences of diploidy in the two instruments. So you could see that we missed 2. Like we couldn't see the same thing in the 30X whole genome sequencing. And this is something we're still investigating. Yeah. So in conclusion, we were able to estimate tumor fractions from plasma samples of breast cancer patients. With both the cheaper approach and the more expensive approach. It's exciting because it increases our confidence that, hey, we could actually use the ultra low pass sequencing to do breast cancer detection in Ghana. And I also read that when such studies are conducted, you actually see lower proportions of people with low tumor fractions. What I'm trying to say is that we could actually use this tool to do detection of breast cancer mildly invasive way, and could help us in treatment. Yeah. So some of the ongoing work is basically to look at the genes in the various regions. So we want to look at the genes which could be found here, which are amplified or deleted. And that's something I'm still doing to the end of the month. And to also determine the sensitivity and specificity of our detection methods. And so for this case, we are planning of sequencing people without cancer, and that could help us really say that our tools are sensitive and specific enough for our population, and to also do breast cancer subtype determination or from cell-free DNA, and it's an ongoing work. I want to say a big thank you to all these people that helped me throughout the way, especially to Dr. Kwatin, my supervisor in Ghana, and Dr. Paz Polak in Mount Sinai, and then Dr. Gavin Ha at the Fred Hatch Cancer Institute, and all the lab members. They've been really amazing people with their encouraging me and providing support at different places, and all my friends that I made here in the States as well. Thanks for all the support. Thanks. Thank you.
Thank you so much, Sam. That was really wonderful.
I just wanted to say a quick hi, Sam. It's good to see you. And we hope to see you in Toronto soon, okay?
Soon. Yes, okay, all right.
In the meantime, let me ask you a few quick questions.
All right.
The first is 90% detection rate of clonal mutations acceptable? And is it possible to improve on this?
Yeah, it is very possible to improve on it. So because we have, sorry, I just want to pull this slide. Because we did the 30x whole genome sequencing, one of the things we are doing is to look at, so which depth, at which depth can we detect tumor? So we are trying to also downsample the 30x maybe to 20x, 15x, and to be able to increase our confidence. That's something we are currently doing.
And would you anticipate different detection rates with different types of cancers?
Different detection rates?
Yes.
I haven't done such a comparison. Different detection rate with different types of cancer. Are you talking of maybe comparing breast and liver cancer? Because these are definitely different cancers, different mechanisms, so it becomes difficult to do to compare both sides. Yeah. But then it has been applied in liver cancer, it has been applied in colorectal cancer, it has definitely been applied in breast cancer. I don't think anyone has done like a pan-cancer detection or analysis yet, but it's been applied in different types of cancer, and mostly metastatic. So you see, high tumor fractions compared to localized tumors. Yeah.
So maybe with regards to that, I can ask you next, what are the most prevalent cancers in sub-Saharan Africa?
Breast cancer is definitely one. Breast cancer is one. Cervical cancer is also one of the increasing. It's one of the top also. For the men, we have prostate cancer also has been one of the most prevalent cancer types in sub-Saharan Africa. Yeah.
And maybe my other question to you as a fellow trainee is, first of all, it must be 3 a.m. or so where you are now, 3 or 4 a.m. Is this correct?
It's 5
Okay, that's not bad. That's not too bad. Well, thanks for being up so early. I was going to ask, how is your ability to change? Because I remember a few years ago, just 2 or 3 years ago, you gave a talk here at the BSI conference on filarial nematodes or some sort of a infectious disease talk.
Great.
This is quite the change of gears doing this sort of like this. Yes. So how was your experience? Maybe can you tell us a little bit about what it took to change your perspective and your skill set so drastically, and how are you finding adapting to the labs at Fred Hutch and Sinai?
Great. I might say it's been very hard and challenging because most of the things I do, I mean, so there is the biology aspect, right? Answering the biological question and formulating your hypothesis, which also takes time and takes some mind. There is also the computer science aspect, learning how to code, learning how to move fast from various systems, working on clusters, supercomputers. Also, definitely take time to learn. Yeah, it's been very challenging. I go home almost defeated every day. That's the
Don't say
feeling.
that.
That's the
That
feeling
means
you
you're
get, when your codes don't work. When your codes don't work. But yeah, I think yeah, but I've also had some great friends and great supervisors, encouraging and also pointing me to places where I could find answers, and that makes me also excited. I'm really excited about this project because it gives me the opportunity to actually apply the bioinformatic tools and learn more. I think there is opportunity for me to learn more
That's really good.
bioinformatics. Yeah.
So speaking of bioinformatics and learning more, I have a new colleague here, Aryan, that I'm going
Great.
to quickly introduce you by webcam. Aryan is a computer science student from University of Toronto. Here he is. And perhaps it's a good time for us to try to relaunch a bioinformatics and maybe coding sort of workshop for through BSI at some point in the summer. If people are interested, we can do a remote workshop and you can both combine experiences to teach people using a platform such as this one.
Great.
Okay. Thank you so much for giving us your time. And with that, I'm going to